the European Center for Atomistic and Molecular Simulation, and we are partner of this European consortium called MDDB. The acronym stands for Molecular Dynamics Database. And together with the other partners in the consortium, in particular with the coordinator, Professor Modesto Orozco, and his co-workers, Adam Hospital and Daniel Beltran, we are planning this series of webinars to introduce the computational community to the MDDB project, which is a pretty interesting project. Is a community project, and so one of the major hallmarks of MDDB is to be inclusive, to try to include as many scientists as possible. And the ambition is particularly high because the idea is to create something very similar to the protein data bank for molecular dynamics trajectories in the field of simulation for biological systems, proteins, lipids, and nucleic acids. Without any further delay, this is the first seminar, the first webinar of a series of about 3 that we are planning to have this year. Without any further delay, I leave the stage to Adam Hospital for an introduction to the MDDB project, and then to Daniel Beltran for a practical presentation about the current version of the MDDB database. Thank you very much, Adam and Daniel, for your time, and please, Adam, the stage is yours.
Thanks, Andrea, for this nice introduction. I hope that you can all hear me well. I'm going to share my screen now. Oop. Okay, so thanks again, Andrea, for the introduction. Thanks to all the attendees for being here today in our first webinar of the MDDB project. Thanks also CICAM to give us the opportunity to start with this series of webinars, showing the work that we are doing in this project and in molecular dynamics, storing of information and sharing the information. So my name is Adam Hospital. I'm working as a research associate in the Institute for Research in Biomedicine in Barcelona, in a small group that is called Molecular Modeling and Bioinformatics, led by Modesto Orozco. And I'm going to give you a brief introduction on the MDDB project. Daniel Beltran is also working in the same group. He's the main developer of the MDDB prototype, and he's going to give you the second part of the webinar, where he's going to show you the current version of the MDDB prototype of the database and the web interface. This is the project that we are going to talk about today. It's the first European repository for biomolecular simulation data. It's called MDDB, and it's a Horizon Europe funded project that was started in March 2023, and it's supposed to long 3 years until 2026, and all of these partners are involved. The IRB in Barcelona, also the Barcelona Supercomputing Center, KTH in Sweden, MBLBI, a small pharmaceutical company in Barcelona called Nostrum Biodiscovery, the University of Oxford, EPFL, and also CICAM, which is the one that is giving us the possibility to run this series of webinars. So first thing to think about is why we started with this project, why MDDB, and for that we need to think about like 60 years ago when the molecular dynamics simulations started. So actually we had previously this year a fantastic CICAM workshop organized in India, where we celebrated the 60th anniversary of MD simulations, the anniversary of this paper that you can see here by Raman. Actually, these are the seminal papers of the MD simulations, the liquid water by Raman here, and the dynamics of all the proteins by McCammon, Gellin, and Karplus. The first papers published about molecular dynamics simulation, about the method. And one point to highlight here is that the first simulation, as you can see here, very small, but I hope that you can still see it, is that the first simulation were run for 8.8 picoseconds. In 50 years, this has changed dramatically. We have increased the size of the system that we are able to simulate. We have also increased the length of the time that we can simulate. Those are just examples. On the left-hand side, you can see the largest system simulated with 1.6 billion atoms that was run by the group of Yuji Sujita in Riken in Japan using the Fugaku supercomputer. Is this system here, but we are now, the community is also running systems like this one, the whole cell simulation by Maring, or the airborne, so the SARS-CoV-2 transfer by the air by Romy Amaro. Very huge systems that now we can simulate, but at the same time we can simulate longer timescales. So as you can see here, for example, in the how fast folding proteins fold paper by D show using the Anton supercomputer, the first time that we simulated 1 millisecond. And this is another example by the Folding at Home initiative by Gregory Bowman and John Codera, using millions of computers around the world and gathering information, an accumulated time of 0.1 seconds of MD simulation. So it's clear that MD has evolved dramatically in the last 50 years. We had an increase of size system by a factor of 10 to the 9. Same for the trajectory length. The ensemble size is something that is also something that we should think about. So it's the number of frames of snapshots that we are storing in each of these simulations. In this case, this has increased by a factor of 10 to the 11. So it's much more frames, and that means size of these volumes of this that we need to store this information. Very important, we have gone from the proof of concept to something that is quantitative. Now people are starting to trust the MD community, the predictions that we are that are the result of our simulations. People are starting to trust this information. And this is a very nice number. So this is taken from the Barcelona Supercomputing Center and says that 15% of the HPC users, I think it's for 1 year, it was used just for molecular dynamic simulations. If we think about all the data that we are generating with all these projects, I think that it's clear also that in terms of data management, we are unquestionably behind. So most of the simulations efforts are lost. So we simulate, we run the analysis, we publish our papers, and after that, we need to think about what to do with this data. Can we store this data? Maybe we have backup systems and we can store this in backup systems, but most of the times the raw data that we are generating with the simulations are just removed after extracting the solvent molecules, and we just keep what we think is important, which is just some of the frames, the most representative frames of the simulation. That means that there is no possibility for a second wave of analysis. We don't have the raw data anymore. There's no meta-analysis possible. There's no gathering of a collection of MD simulations, MD trajectories that we can analyze at the same time. There's lack of reproducibility and quality checks. This is really important nowadays. Most of the journals are starting to ask for reproducibility of the results. That means that the raw data, and not just the raw data, but also all the protocols followed to set up the system, to run the system, which is the MD engine that has been used, what are the parameters that we use for that. All of this should be stored somewhere. Of course, there's no AI learning on top of the data. We are in the era of data. If we accumulate this data that we are generating, huge amounts of data in MD simulation, we could use AI on top of this to train models, as you've seen in our field lately. And then there's a poor interaction with other fields. So all this flexibility information that we are extracting from this data is not integrated in the life science ecosystem databases. So we have many databases that we are using every day, and there's no information about flexibility. There's barely information. At least all the one that we are extracting from our simulations is not there, and we think it should be there. So there's a question that has been on the table for many years now, and is it worth storing these MD trajectories? What would happen if we store this information and we open this information to the community? And for that, there's a very nice example that we have been involved for many years now, and this is the Asconaby DNA Consortium. This is a consortium by many partners working in MD simulations in the field, and with a specific interest in DNA simulations, with one clear main objective, which is to collect data to describe sequence-dependent properties of the DNA. This consortium started in 2001, and basically what they did is to gather together and in a way, using this divide and conquer regime, they distributed these 39 MD simulations through all the different partners, and they simulated 2 or 3 simulations each of one, 40-mer DNA structures at that moment, 100 nanoseconds length. This project keep ongoing with different rounds. So the second round was micro ABC, 1 microsecond. Third round, 2019, from 39 MDs to 52, and the last round is still running now. You can see the jump from 52 MD simulations to 380 MD simulations, and now 10 microseconds. This is what we are simulating now. What this initiative is trying to extract, as I was saying before, is just the sequence-dependent properties of the DNA. So we are analyzing this base pair step. We are extracting helical parameters, so the flexibility patterns of this base pair step. And we want to see if these flanking nucleotides here in the tetramer or hexamers are affecting the flexibility of this base pair step. This is the main objective. This was the main objective since the beginning. But what happened with all the data that we generated and we opened to the community? This is the reality. The real output of this data is more than 100 papers, and this is still running. These papers are citing this data that the consortium is generating. That means that this data is important, is really important for the community. The community is using the data if we open this data. So it's everything about data, and in our case, about MD data in particular. As it happens with all data and all the studies nowadays, the amount of data that we are generating is growing exponentially. And again, it's been a matter of debate in the field if the data that we are generating can be considered big data or not. I think that if you see these keywords that are the ones defining big data, we have all of the keywords, so we are generating data in a very in a fantastic, in a fast velocity, in a fast way, especially since we have GPU cards that we can use. Variety, we have many different formats, trajectories, topologies, analysis, volume. Trajectory files are very big, as you will know, as you all know. If you think about all the system with water molecules surrounding the system. And also complexity. We typically run the simulation in supercomputing centers, and then we need to transfer all this data from the supercomputers or the data centers to our machine. Most of the times we cannot download all data because it's too much. We need to pre-process the data, so it's really complex. And so we wrote a review about that 5 years ago, and we were kind of right on the predictions, although for us, it was not expected the real growth that we have 5 years after that. So if you think about what we are doing in our field, in all the research groups, we are not so far from the data-driven process. This is a data-driven pipeline. If you want to, let's say the one that we are doing in biomolecular simulations, what we are doing is to collect the data, as you can see here. So we are generating MD simulations. This is the raw data that we are collecting. We are reporting the data. That means that we are organizing the information. So the system that we have simulated, it is linked to a particular protein. We know the name of the protein. We know the link to the PDB file. We know the UniProt sequence. We know the link to all the different life science ecosystems. So this is organizing the data. And then, of course, we are running analysis. That's the main objective for running the simulation, is to extract information from the analysis. And we have all of this. The only missing piece of the data-driven process is to open this information to the scientific community so that the scientific community can take action on this data and extract added value. Is the only missing piece. we've been working with this for years and years in Modesto's group. We started in 2010 with the first MDDB MD database that was called Molecular Dynamics Extended Library, that was monomeric proteins. We continued with the nucleic acid specific database, then a small molecules database, then protein membrane database. When the pandemic appears, we jump to the COVID-19 related protein simulations. And after all these years, we are really happy because finally we have convinced the European Commission to fund the first European and generic MDDB database. And this is exactly why we are giving this seminar today. The MDDB project has 5 clear objectives, which are the ones listed in this slide. So the first one is to set the foundations for the adoption of FAIR principles of molecular simulation data. FAIR principles are everywhere, as I'm sure that you know. We are going to try to apply these principles for the first time on MD data, on MD simulation. So we are trying to move to the open science paradigm. We want to promote the full and useful integration of molecular simulation data on the life science data ecosystems. Again, this is what I was telling you before. We need to integrate the flexibility data that we are extracting that we think is really important in the life science ecosystem. We want to define and promote good practices for the generation and analysis of trajectories. We still don't have any kind of a standard in our field. We don't have any protocol for pipelines to run simulations. This is something that we think that we can define working with the community. We want to design the technical infrastructure for a general repository for simulation data. And for that, we have decided to go to a distributed platform. And this is something that Danny will talk about in the second part of the webinar. He's going to show you how we are planning to develop this distributed platform. And he's also going to show you the prototype and the web interface that is on top of this platform. And finally, we want to design a sustainability and governance model for MDDB. So we need to ensure that this distributed platform that we are now starting to deploy in many different centers, especially HPC centers, can be sustained and can be maintained in the near future. The second part of the webinar is focused on this one. We will have different webinars on this series of MDDB, but now Danny is going to show you the distributed platform. So this is going to be the MDDB prototype. And now I'm going to stop sharing and giving the floor to Danny.
Thank you, Adam. I will share now the screen. So allow me to go full screen. Can you see it properly? Okay. So now I will explain a few more technical details about the current state of MDPosit, our prototype. As Adam just mentioned, this is a federated, we are trying to become a federated database. This means it is a decentralized database. Every research group may have a different node, and they have full freedom to upload or delete data from their node. Once data has been loaded to the node, they also have plenty of freedom to publish or unpublish their simulations from the global server so everybody can see them. And they may have also hidden simulations for themselves. Also, I would like to mention that since it is a decentralized database, a node may be anywhere physically. So a research group may have its own node at home. For the first part of the talk, I would like to focus in a single node, how a single node works. So let's forget for a moment about the global system, or the federated system. One node, it's a database, an independent database on its own, which means that even if the global server has fallen, the node can keep working flawlessly. It does not depend on the global server at anything. So let's go to have a overview of how this independent node works. This will be a very brief, very simple scheme of how it works. It's also like a data pipeline, since we start working with the input data until it is available for the users. So in this case, input data would be just a set of available MD simulations. And the first step is what we call the workflow. The workflow is the one who prepares data to be loaded to the database. I would like to explain briefly what we do. The workflow is a software which has multiple roles. The first role is gathering metadata. We gather different types of metadata. We have titles, authorship, references, things that will help you find a simulation when you are browsing this actual simulation. We also gather several MD parameters. We have time step, force field, and long, etc. And here we try to be also flexible. So we think it's an interesting debate to bring to the community what metadata should be gathered, what metadata should be kept in the database. So the second role of the workflow will be in quality control. We test data to make sure it is coherent. We look at very obvious things right now. We check that the topology has no errors. For instance, we check that the number of atoms, a number of bonds for every atom is coherent. We check that there are no sudden jumps in the middle of the simulation, these kind of things. But as it happens with the metadata, I think we think it is interesting that the community tell us what is important to check, what tests should be done for the different cases, different systems we may encounter. So again, we are flexible. And the last role of the workflow is analyzing MD data. So generating these analysis results, we will also load to the database. Here in this image, there is a summary of the analysis we have right now in the visual available in the visual interface I will introduce later. And we have very generic analysis, and we have system-specific or system-oriented analysis. And this list of supported analysis has never stopped to grow. We implement new analysis as we support new systems and as more authors request a specific analysis. The same that with the other two roles, we are flexible, we are open to keep implementing new analysis. Once data has been prepared by the workflow, we are ready to upload this data to the database. Here I would like to mention that we use our own software to manage, loading, deleting, updating data in the database. And the only thing I would like to say here is that we have our own in-house system to store atom coordinates. And this system allows us to retrieve specific atom coordinates for specific atom selections at specific frames in a very efficient way. It is very fast. This database is powered by MongoDB, which is a state-of-the-art NoSQL database. And once we have uploaded our data to the database, it is ready to be accessed by the user. It is already available. There are two ways for users to access MD data. We have the programmatic interface and we have the graphical interface. The programmatic interface works through simple HTTP requests. It's something you can do using a terminal or using a web browser. And it allows you to browse simulations, download atom coordinates, download analysis results, download files, these kind of things. It is fast, it is efficient, and it is fully documented. So for data scientists, it should be a comfortable experience to mine data from the programmatic interface. Then we also have the graphical interface, which is more user-friendly. It allows you to do most of the things that the programmatic interface will allow you, but it also includes a molecular viewer, so you can actually see how the simulation is moving. You can see the structure with actual coordinates. And it also allows to better represent data. So instead of downloading the round numbers, you can have graphs, you can have heat maps to better understand what data is telling us. So now I will go through a very brief tour in the web client, which is the graphical interface. To access the graphical interface, you can access this URL you are seeing highlighted in the screen. So the first page you find when you jump to this URL is the home page. And here you have a search bar, or if you click to the browse button in the header, both ways you go to the browser. Here I've prepared a simulation I would like to show you. This simulation is of a nucleosome, which is a mix of protein and DNA, which is interesting as a system. What you see is a summary. So you have the accession number, which for this specific simulation, which is unique and persistent. These are two of the principles of the FAIR rules. We are trying to be as compliant with the FAIR rules as possible. Then you can see the title of the simulation, a preview, and a summary of the available analysis for this specific simulation. Because available analysis may change from one simulation to another depending on the system. So if we click in this simulation to have a closer look and start exploring, one of the first things we will encounter is the molecular viewer. So as you can see, it is not a video. It is an actual viewer. It contains the coordinates, so you can zoom in, zoom out, turn, twist, these kind of things. We also have plenty of features, so you can customize how you want the simulation to be displayed to better understand. And I would like to say also that this molecular viewer, it is not showing you the whole trajectory, which is in the database. In the database, we have the whole trajectory, but in the web browser, in the web client, we only have a summary of the trajectory, because if you try to load the whole trajectory, your web browser may explode because it is too heavy for the memory of most computers. We only represent a small subset of frames taken along the whole trajectory. If we keep exploring, we will soon find also the analysis menu, which it is the door to all the available analysis. In this case, I would like to show you the RMSD analysis. It is a very common analysis you all may know. In this example, we can see in the graph that we have RMSD value for several frames along the trajectory against the first frame. For this specific system, the nucleosome, we have computed separately protein and nucleic acid RMSD values. And in the headers above, you can see we also have available the RMSD against the average structure or the TM scores, but now we will focus in this RMSD analysis. This is a good example of the benefits of having trajectory data next to analysis data. Imagine that here you see a big peak and you want to check what is happening, why there is a sudden rise in the RMSD. You could click on the graph, and then the viewer would appear and will show you the structure with the coordinates of the specific frame you just clicked in. So it is a powerful tool. Another example in the same line, another analysis I would like to show you is the hydrogen bonds analysis. Here I would like to explain a bit how the interface works, because it is not easy to represent this data. We have redone this interface several times to try to make it as user-friendly as possible. The three columns in the left are telling you which are the atoms which are participating in the actual hydrogen bond. So in the left, you have the histone octamer, which will be the protein, the center of the nucleosome. In the right, you have the DNA, the DNA wrapping the nucleosome, and in the center column, you have the hydrogen atom. Also, you can see if this atom in the center belongs to the protein or to the DNA. You can see which is the donor. In the right, these bars you see, these horizontal bars, they represent whenever the hydrogen bond is happening or not along the trajectory. So gray means no, blue means yes. I would like you to focus in this specific bar I'm highlighting with an arrow. I think it's interesting because it's an hydrogen bond which is not happening at the beginning of the trajectory, but then suddenly appears and it is very stable until the end of the trajectory. In this case, if I see correctly, it is nitrogen from an arginine 40, which is interacting with an oxygen or the phosphate group in a 1N. And the donor is the arginine. Okay, so as in the previous analysis, what happens here? That we can go and click in the bar, so the viewer appears, and you can see this beautiful hydrogen bond, which is not happening at the beginning. You can see it very highlighted, and you can see dynamically how the hydrogen bond is formed through the end of the trajectory and stays there. So what data was telling you, you can actually see it, and it is very explicit. One more analysis I want to show is energy analysis. Now we have changed the system, so forget about the nucleosome, not a nucleosome anymore. Now we have a protein-ligand system, like in the picture you can see in the left of your screen. And this analysis is a good example of the system-oriented analysis I told you before. We designed this analysis for proteins and ligands, although then we have ended up using it for other systems. This analysis is consists in computing the electrostatic and van der Waals energy interaction between the protein and the ligand along the trajectory, and then we made an average value, and that's what you see here, the energy for every residue in the protein. And with a very fast overlook, you can identify which residue is carrying the interaction, which residue is having the strongest bond, the strongest interaction. Here you have a very negative peak, and again, no surprise, if you click in the peak, you see the residue in the structure, which is very close to the ligand. In this case, it's an histidine, and this residue is the most important probably for the interaction, as predicted by this analysis. The last analysis I would like to show you is this analysis, which again is very specific, and we are changing of system again. Forget about the protein with the ligand. We are moving to a spike, the SARS-CoV-2 spike, this virus you may have heard about. Why is this analysis so specific? Because I don't know if we could call this an analysis. It's actually more a tool. What we have here is a representation of the entropy, or how prone is a residue to be mutated, in the spike, both in the structure you can see above, yes, and in the sequence you can see below. Red means high probability of mutation, and white-gray means low probability. So this data has been mined from the GSA database, and we can represent it in a very visual, user-friendly format. This will be the last analysis I want to show you today. Time is limited. So now let's get back to the idea of the global system, of the federated system. I was explaining you one of the nodes, but of course you don't have a federated database with one node. We need several nodes. I will briefly explain the nodes we have now. So far, we have 3 nodes in Barcelona. The 2 above are in the IRB, where we are from, and the one below is in the BSC, in the Barcelona Supercomputing Center. And last week, we traveled to Oxford, and we set up our first international node. These 4 nodes are already connected to the global server as a proof of concept, and it is working just fine. So that's how things are now. And what is the global server, you may ask? The global server, at the end, for the user, it is just like another node, let's say. You have your web client, your graphical interface, you have your programmatic interface, and for you, they behave as any single node. Like you can see all the simulations together in the same browser, and you can enter any of them. It is as fast as a node, although in the back it is weightless. Okay. So last thing I would like to explain, this concept has been introduced by Adam in the previous talk. The concept of meta-analysis, which very briefly will be like, I'm not analyzing a single simulation, I'm analyzing a set of simulations at the same time, so this has a higher statistical value. I would like to explain that we think it is one of the most powerful advantages of this database. Like you have a very strong programmatic access connected to a lot of simulations, not only simulation in one group, but simulations around the whole world, because it is a federated database. And it is very interesting what analysis could be done. Here we have prepared a few examples. We could, for instance, analyze active site and the shape, the size, the composition, and ligand interactions from a collection of protein-ligand MD simulations to explore change on flexibility patterns in the active sites upon structure modifications, such as mutations, additions, deletions, on a collection of protein-ligand simulations from a specific protein family. Another example would be checking Watson-Crick hydrogen bonds behavior in guanine cytosine versus adenine thymine pairs from a collection of canonical bDNA MD simulations. That will be to explore the propensity for GC or AT base pairs to show frying or hydrogen bond opening processes from a collection of canonical DNA simulations. This is something we are currently analyzing in the ABC project that Adam mentioned previously. Another example would be salt bridges generation and residence times by residue type. This is interesting because this meta-analysis could be performed on the whole database. It is very generic. And it will be interesting to track statistics about salt bridges formed on ionizable amino acids, including propensities and residence times. In my last slide, I would like also to explain that since we think meta-analysis are an important thing, we have prepared in the graphical interface a page which is dedicated only to meta-analysis. We have provided examples, tutorials on how to do this. As a tutorial and as a use case, we prepare Jupyter notebooks which run in Python. You can, using the API, you can do whatever you want. In this case, we have a subset. Okay, let me explain. This is the first use case we prepared, and it was in the time of the COVID pandemic. We have a family of simulations. There are 78 simulations from DSO of the human ACE2 protein interacting with different ligands, which are FDA-approved drugs. And since we were running our specific analysis of energies, it was very easy to mine this data from all the simulations at once using the programmatic interface. So it was relatively easy to create this graph you see in the right of your screen, showing these interactions, the energies, and you can easily identify which ligand is performing the best, the strongest interaction according to this prediction, which will be the one in the top. I don't know if you can read it, but it is called Ruthasbeer. So now that you know which is the most interesting ligand, you can create this graph in the left showing the energies analysis per residue as it was done in the web client. So instead of using the default graph we provide in the graphical interface, you can build your own graph as you prefer using Python. And it is very easy to do. And that will be pretty much everything. Thank you for your attention, and I'm happy to take any questions. Thank you.
Thank you, Danny. If you have questions, you can open the mic, you can write in the chat. I don't see any hand raised. Oh, yes, one, sorry. No, it was there, but it's not there anymore. Here. Okay, so there's one question from Milana. Milana, if you want to open the mic, but the question is, how is one granted a note? Oh, they can. Oh, okay, sorry, I didn't know that. So I guess I can answer this question. So most of the things that I presented in the introduction and that you can think about are on the table now and are ongoing discussions on the MDDB project between all the all the partners of the consortia. And we also want to open these discussions to the community. So this is one of the discussions that we have. So we need to think about where we want to deploy a new node. What we are doing now is to deploy new nodes in in internal partners. You've seen the Oxford example, but we are also exploring HPC centers, which they have data centers. And I think that that's convenient because if you think about the data-centric regime, we should move to where the data is, and we should not transfer all the data up and down. So we are now, actually, this webinar, Danny and myself, we are in Cineca, in Bologna, in Italy, and we are trying to deploy a new node here. So it's still on the table, but we are thinking about the HPC centers, also country-related nodes. But as Danny has mentioned, this infrastructure is easily deployable even in a small research institute. So this is another possibility that we are also discussing internally. There's a second question by Gianmarco Rigagnese from Optimate. Is how do you see the role of Optimate with respect to MDDB? Maybe Danny wants to say something about this.
It is an interesting question. We have been discussing also internally this part. Right now we have this implementation we made with you will remember, Gianmarco. We implemented Optimate in one of the services. It should be available as well now, although I have not checked in depth, so better not to try. It may explode. But yes, MDDB, I think, is strongly trying to get compliant as much close as possible to Optimate. So I think there are also, we are planning, I don't know, because there are politics here. I should not say anything. So we have another question here. Are we able to start getting API access to trajectory data? Yes, the API is already open. So I must say, since we are still playing, since we are still in a very early stage, at least I would like to remember this is a prototype, so beware. The global server, it is still working, but we only have published like a few simulations to play. However, if you go to one of the nodes, in specific my node, our node, I will copy right away the link, so you can have it in the chat. This node is the one who has more simulations loaded right now. Okay, I just copied the link in the chat. This node has a lot of simulations. It is the most populated node, and it should be more stable than the global server these days that we are start playing. You can access the, actually allow me to copy to you the API URL, so you can jump straight to the documentation. So here, if you click in this second link, you will be you will be taken to the Swagger page. Swagger, you may be familiar with Swagger, is this standard documentation system for APIs, for programmatic accesses. So you can see all the endpoints, all the possibilities of the API, and start playing right now if you want. It is already open. So thank you for your question. Next question is, hello, Danny and Adam. Nice talk. Is there a limitation of the size of the dataset we can put on the MDDB, like hundreds of gigabytes? That's a nice question. I think this depends on the nodes. The idea of having a federated database, one of the main advantages is that every owner of a node is free to do what they want. So if you go to a node in a very strong computational center or a very strong group which has a lot of disk, they may be happy to upload all your data. So yeah, the technology has not a limitation by itself on the size of on the weight of the data you can add. It is more than you have enough disk to store it. So that would be my answer. I don't know if you agree, Adam.
Yes, we are still exploring the possibility for example, upload in a way that then there is very efficient, the extraction of the information is very efficient, raw trajectories, meaning all the systems, including water molecules. This is something that is the most complicated part. Right now, what we are doing is just to extract and dehydrate the system before extracting the water molecules. This, of course, is reducing the size and the amount of information that we need to load into the database. And this is working really well. This MongoDB in a distributed infrastructure with many machines working in parallel is giving a really fantastic efficiency. But we still need to explore the next step, which is storing all the simulation, and that is hundreds of gigabytes. This is the next step.
Next question is, hello, I wonder how we can upload the data to the database. Thank you. Yes, this is probably the question we were most afraid of. Well, right now there is not an established protocol, and this is something we would like to discuss in the MDDB consortia. But I will tell you how it works right now. Right now, all this backend part I show you in the slides, the workflow, the database loader, is something that every node has its own, and only the people who are there can access this software. So the idea is that if you want to publish something in a node, load something in a node, you have to reach out with the owners of the node. You have to send them data. We don't have also a strict protocol for this. It could be an open repository. It could be a hard disk. And then they can start playing with your data. Right now, we try to make these tools, these workflow tools, as user-friendly as possible. So people who is having this role of keep the maintenance of a node or myself, we have a easier job to do it. But this may change very soon because we have several plans to make this process more user-friendly. So this may change very soon. Yeah, that will be my answer.
And this is quite linked to the next question, which is what are the expectations from the node? So please remember that what we have shown you today is a prototype. So this is, we are just exploring the possibilities. Is the distributed infrastructure working well? Is the master taking all the metadata and showing information through the API on all the different nodes? And this is, it seems to be working really well, but it's still a prototype. So the expectations on the nodes now is just to try to maintain these prototypes. The expectations on the nodes, once we go into production mode, is something that, again, will need to be discussed within the consortia. But of course, you can think about sustainability, maintenance. We don't want a node to be there for just 2 years, and then after that, something happens with the data volume and there's no data available anymore. We need some kind of sustainability in all of these nodes. But again, this is something that we'll discuss. And this is why we are thinking about HPC and data centers, actually. So next one is, how do you think you can deploy your analysis, like H1 analysis, on very large systems where you do not know a priori which part of the system is interesting? Maybe Danny?
Sorry, let me reread.
So the idea is you don't have what Danny called these agents. So each part of the system that can interact with a different part, and is calling these agents or fragments. So there's now there is a form where the users can just input which are the fragments that you think are the important ones, and then we are computing hydrogen bonds, energies, etc., between all the different fragments. If you don't know these fragments, we still don't have a way. So we try to gather this information automatically from the system if the user is not telling us, but we give the user the possibility to define explicitly what are the fragments or agents. So this is what we are doing now. Comments and suggestions are welcome, Matthew.
I'm having a delay. I don't know if you are having a delay. Oof, I'm having an extreme delay. I cannot.
Next one, because we are reaching the top of the hour and we have many questions. So Adam, have you checked the quality of existing data that you include into MDPosit? So this is something that, again, is on the table on the discussions within the MDDB consortia. The ones that we have now, we have this standard quality check that that Danny was explaining before, in the number of bonds per atom, in the number of atoms per residue, in the jumps between the frames in the trajectory. These are the ones that you can think about, but nothing other than this. With metadata, you were able to collect a set of MD related to H2 protein and run analysis on the whole set. Yes? Did you do this from the website? No, from a local environment. From a Jupyter notebook, actually, you can do that using the programmatic access, as Danny was explaining. So you have a Jupyter notebook available in the website if you want to play.
I will try to show to share the link to this specific. Yes, here it will be. Let me copy it in the chat.
Okay. In the meanwhile, how accessions are built, and are there any special rules? So this is a good question. Now we are working on the persistent IDs by ourselves. That means that we have persistent IDs for each of the nodes, and then we have one global persistent ID for the master that is connected to the persistent IDs of the nodes. The way that we want to go is to try to give a DOI for each of the simulations uploaded into the MD database. But it is something that, again, needs to be discussed. I think it's the best approach, but it requires paperwork. We need to be granted so that we can grant one of these DOIs, and this is not easy, but is the way that we are exploring now.
Here I would also like to explain that accessions, as they are now, they are, well, we read a few papers of good practices when generating new accessions, and we have followed them as we could. But at the end, in the local nodes, we have also granted some freedom. So if the user who is about to upload a simulation, they want to use their custom accession because it makes more sense for them, they can. And of course, if they fall into having repeated accessions or something like this, they should not be able to do this because this system that generates the new accessions has its own systems to tell the user, hey, beware, you are going to duplicate an accession, or these kind of things. So this is it. Thus, the central node is aggregating the data of all other nodes in the research panel to have a single point of research. Thank you. Yeah, that's a question I wanted to hear. What the central node is keeping is only the very basic metadata of all the simulations. So it is keeping, the title, authors, these kind of things. So when you try to do a query over the available simulations, the one who answers is the global server by itself. He doesn't have to go querying any node. But in the moment that you request atom coordinates or an analysis or a file, or try to download a file from the global server, it redirects you. It is a HTTP redirection to the corresponding node under the hood. You don't even have to care about it. It is done automatically. So yeah, we are not duplicating data. That's why I said very briefly that the global server has its own database, but it is weightless. It is almost has nothing, no weight almost.
Next one is also for you, Danny. How is multiple replication of one single simulation handled?
Okay, yes, it is something I didn't mention, and I think it is better if I just show you. Let me share the screen again very briefly. So yeah, I think here you can see my browser also with the header, but whatever. This is a live demo again, so well, beware. Things may explode when you do things live. So let me think about an example with multiple replicas. I think this here. Yeah, this is the spike I show in the presentation. It's a simulation from Amaro. It has, as you can see here in the browser, it has 3 MDs. Instead of calling it replicas, we call it MDs because we try to be also flexible with scenarios where, let's say, we have several trajectories for a single topology, but trajectories maybe are not replicas, but they are something different. Some example of this, well, I think it's too much if I give examples because I may step into things I don't fully understand. So yeah, let's say this is the way we handle replicas, but it is flexible to handle other similar things. When you click inside here, you have these buttons here to switch to one replica or another. And if you note in the URL, what has changed is this 0.2, 0.1, 0.3. This is what takes you from one replica to another. So if you are in another subsection of this simulation, you may manually change the URL to jump to another replica. So yeah, that's how we do it. So we don't have to replicate all the topology data if it is topology and other data which is identical for the replicas. I hope this answers your question.
Okay, I would like to thank the audience because we had many questions. This is fantastic for us. And sorry, Odd, if we are running out of time. I hope that this is not a problem. So if it's a problem, just please stop us. And we are going to try to answer the questions that are still on the query and answer chat. So from Bala here in Chineca, he says compliments for the nice work. He can ask us directly because we are there in the same room, but he wants to ask this as it may be interesting for others as well. Do you have any roadmap to make this platform a sustainable one, but in particular about the platform's technical sustainability? So will you be able to convince journals to consider MDDB as alternative to Zenodo? I think that this kind of infrastructure or the project that we are building, it's much better than just uploading a simulation with just the files as you might be doing in Zenodo. It's not that just it's not just that you may be able to download the trajectories, you also are able to see the trajectory, to take a look at how the trajectory, the system is moving. You have the analysis, you have the interactivity, you have the programmatic access, not just to the whole trajectory, but also to just fragments of the simulations. So it's much more than just one link. And if we succeed on having a DOI for each of the simulations, I think that this is the way to go. And this is what we will try to convince the journals, the commission, and also the scientific community. And I guess that for that, we also need you, the community, to support us. And this is why we are starting this series of webinars, and we will be disseminating this in this project on and on. Okay, next one is from Mark Baden. Thank you for this update on the project. I wonder how the analyses work. Does the user do them, or is the database, once it gets a new data set running, a predefined set, or are they done on the fly? Do you want to answer that, Danny?
Sorry, I was muted. No, most of the analyses are pre-computed and stored in the database, so you can access them. That's why you can access them so fast. If you want to run new analysis, it is something we consider it should be done through the API using your own cluster. So we don't run almost anything on the fly.
So yes, the idea is that we have a pipeline, we have a workflow, and the workflow contains all the analysis, the quality control ones, and also the energy, the pockets, and all the analysis that you can see in the interface. Now we are running all of this. Every time we try to upload a new trajectory, we are the ones running all of this. In the project, there's something written about the possibility to deploy a virtual research environment open to the community so that the community have direct access to this workflow, and you can upload your own trajectory, run the workflow, and obtain the results. This is something that are on the plans, but it's still not there, and it's something that again will be discussed during the projects. Also, in terms of working with the data, do you use an ontology for semantic relationships? I think I can answer something about ontologies. We've been working, as you well know, Mark, together with BioExcel on trying to extend the EDAM ontology, the one that is used in Elixir, basically. It's a bit complicated. We managed to add new terms related to MD simulations, but at that moment, most of the terms were rejected because they were just used by one single tool. So imagine, I don't know, TPR files, which are the ones that now you can read TPR files with MD analysis in this kind, but at that moment it was so specific for GROMACS tool that was rejected. I don't know if EDAM is the one. We still need to think about that. There's one particular work package on the project that it's dedicated to standards, formats, and metadata, and also ontology terms. So it's something that we for sure will be discussing during the project. So next one, could you have some kind of clustering of the structure for these fragments based on coordinates or fluctuations? Dani?
I think it is the next analysis in the list. One of the very next analysis we have think. I suppose you mean if we have clustering based on coordinates or fluctuations, or if you have if we have also the fluctuations analysis. I don't know, because we have a graph with atom fluctuations, which is probably not what you mean, right? I don't know. Well, we can see it in the web page very fast if you want. I don't know. We maybe are running out of time.
We don't have time for that, Dani. Sometimes it's complicated because we cannot have a direct communication. But we have pairwise analysis now, where you can more or less see the different clusters, different conformations on the trajectory. We have PCA, where you can also identify conformations. We are now working on the clustering, just coordinates clustering, Cartesian coordinates clustering. So there will be many different ways to analyze the different conformations of a system in the database. Okay, next one, are you planning to impose a general rule on the simulations, like number of replicates, choice of force fields, length of the simulations? We are not planning to impose anything. What we are planning to do is to work on best practices. So within the project again, and then with the community, we want to try to write a set of best practices, not just on the setup of the systems, also on the running of the system, also on the provenance that we need to store, also on the metadata we need to store. And for example, the choice of the force fields will be there. The length of the simulation, that depends a lot on the system and the study that you are that you're starting. So trying to summarize, we are not going to impose anything, but we will try to write a set of best practices for this. And it's good because one of the partners is KTH, so the main MD developers of GROMACS is helping us a lot with this. Last question, it might have been answered, sorry if so, but is there a way to filter out trajectories entries based on biomolecules included? For instance, I put just one trajectories with RNA only. I think that this is available from the search or the advanced search, Dani?
Yeah, we have different search. I mean, maybe if you saw in the header, there was like a search page. You can enter and you can filter by many structural features or so filtering DNA specifically. Right now, I should have to check, but there should be something to filter DNA. Right now, I will have to check, and we are a bit out of time. Of course, if it is not there, we have to implement it very soon. So yeah, you can filter by other parameters which are indirectly telling you that it is DNA, but yeah, it is not as straightforward as filtering by protein DNA. Yeah. We definitely should implement something like this.
Noted. Thanks for that. Okay, it seems that there's no more open questions. I guess that we don't have time for more. I just would like to thank again all the audience for being here with us today. And please keep in touch with us. Take a look at the website. We are going to run different webinars like this one in the MDDB series together with SICAM. So please follow us. And you also can, I think, go to the website and see all the information. We will try to keep up to date the website with everything that we are doing in the project.